I'm happy to introduce our speaker, Tessa Firestone, Research Associate. So Tessa's earned a Bachelor of Science from the University of Pittsburgh, and after receiving her undergrad, Tessa worked at the University of Nebraska Medical Center, investigating drug-resistant mechanisms in the malarial parasite Plasmodium falciparum. So Tessa's been in her current position as a research associate at EpiCypher since November 2021, where she's contributed to numerous developmental and collaborative projects optimizing and utilizing epigenomic technologies such as CUTANA CUT&RUN, cut and tag. She's also contributed to enhancing the understanding of chromatin interactions and their impact on therapeutic discovery. So with that introduction, I'll hand it over to Tessa to share what she's learned optimizing cut-and-run.
Thank you, Nathan. So today I do have the pleasure of discussing CUTANA CUT&RUN, one of our genomic mapping technologies. And by the end of this presentation, I hope you will all be able to understand the fundamentals of the cut-and-run protocol, learn to navigate selecting antibodies and identifying the best reagent for your target, master the preparation for cut-and-run-ready samples, and getting the best practice of data analysis. But before I dive into the protocol and our user suggestions for achieving a successful cut-and-run experiment, I want to take this time to briefly introduce everyone to chromatin. Chromatin refers to the structural organization of genome in the eukaryotic cells. And within chromatin, you have nucleosomes. Nucleosomes are a complex comprised of DNA wrapped around an octamer of histone proteins. And these nucleosomes are the building block of chromatin. Chromatin is essential for cellular processes, including gene expression, cell division, and DNA repair. These protein nucleic acid assortment is regulated by a diverse modifications in proteins. Some modifications include methylation and acetylation, as well as DNA methylation, and chromatin-associated proteins, including transcription factors, remodelers, chromatin readers, and erasers. But since chromatin is modified to alter cellular processes, dysregulation in chromatin features can cause disease. And this is where genomic mapping becomes a useful tool to reveal the relationship between chromatin and disease. And when we talk about epigenomic mapping, we're talking about identifying the chromatin modifications in the proteins associated with DNA across the entire genome. Mapping reveals genes that are being repressed or associated with activation in different cells, different diseases, or under various conditions. There are different assays available to create these epigenomic maps. Historically, genomic mapping relied on technologies such as ChIP, and the standard ChIP workflow followed these key steps. Cells were cross-linked, and this is where proteins are fixed to the DNA. And then chromatin is often fragmented, either by sonication or cell lysis. Target antibodies were then used to immunoprecipitate or pull down the protein DNA complex. And after a series of washes, libraries are prepared for sequencing. ChIP has led the field in exploring transcriptional regulation and chromatin dynamics. However, ChIP-seq has significant limitations. First, ChIP-seq requires a high cell input. It needs millions of cells to generate a sufficient signal, which can be challenging for rare populations or clinical samples. ChIP-seq also requires deep sequencing depth in order to overcome the low signal-to-noise ratio. Cross-linking and chromatin fragmentation can lead to non-specific DNA pull-downs, increasing the background signal. ChIP-seq was foundational in understanding the genome. However, it is extremely limited with the high input needs, background noise, and the resolution constraints. And with that in mind, let's look at one of the next generation techniques, cut-and-run. Cut-and-run, which stands for cleavage under target and release under nuclease, is a revolutionary approach that is sensitive, robust, and can be easily scaled for large experiments. In this approach, cells are immobilized on a magnetic solid support. Cells are then permeabilized and incubated with a target-specific antibody that is fused to a protein AG is added. And this protein is responsible for the selective cleavage and release of the antibody-bound chromatin. Cells remain onto the solid support while the cut-and-run DNA is purified from the supernatant, and this DNA is then further processed through library prep and then sequenced using next-generation sequencing. Cut-and-run offers improvements in throughput, sensitivity, and cost over ChIP. Some highlights include the required cells. Each reaction starts with 500,000 cells, which can go down to 5,000 cells depending on target. The lower sequencing cost. Cut-and-run data only needs a fraction of reads compared to ChIP, a total of 5 to 10 million reads per reaction. Cut-and-run has a high signal-to-noise ratio with few stringent washes, and this results from the direct sequestration of the target-enriched chromatin. And to further increase experimental throughput, cut-and-run has been adapted for use in H-strip PCR tubes. This enables streamlined processing, meaning the user can easily process over 24 reactions in a given experiment, and this increases experimental reproducibility with the use of the multi-channel pipettes. Now, let's take a side-by-side look of how CUT&RUN compares. So these are genome browser tracks for two targets, H3K4-trimethyl and H3K27-trimethyl, from a ChIP-seq dataset in gray and a CUT&RUN dataset in blue. You can see narrow punctate enrichment at promoters for H3K4-trimethyl. And then we can see broad enrichment for H3K27-trimethyl for both datasets. But when we go into the experimental details, we can see the advantages of CUT&RUN. Starting first with sample input. ChIP has used fragmented chromatin, where CUT&RUN uses intact cells or nuclei. Next, looking at the number of cells, the ChIP-seq has needed over 1 million per reaction, where CUT&RUN needed 500,000 cells. Next is the sequencing depth. ChIP-seq needed over 30 million reads to create a sufficient signal, where CUT&RUN only needed 3 million reads to generate the same enrichment patterns. Next is the signal-to-noise ratio. In the signal-to-noise ratio, it refers to the ratio of the meaningful biological signal to the background noise. And a high signal-to-noise ratio means that the peaks stand out clearly from the background, making it easier to interpret the data with confidence. CUT&RUN addresses many of the limitations and offers high resolution with lower sequencing cost and fewer cells. And this CUT&RUN throughput allows the user to expand their experimental design and examine more targets in different cell types. So next, I would like to take a moment to walk through a few more datasets generated using CUT&RUN. These examples will highlight the versatility of the assay across different targets, different cell types, and experimental conditions. So first, we are visualizing CUT&RUN tracks for various histone PTMs and transcription factors, which highlights the patterns of co-enrichment and regulatory architecture. Within these genome browser tracks, each row represents an antibody tested for a particular target. The peak height represents the enrichment of each antibody tested. And this allows you to compare where you see enrichment for different chromatin features, such as enhancers, promoters, and transcription factors. But we could also use CUT&RUN to explore how the distribution of histone marks varies between different cell types. Shown here are genome browser tracks for 3 targets: an IgG background control, H3K4-trimethyl, an active promoter mark, and H3K27-trimethyl, a repressive mark, across 3 distinct cell types: granulocytes, lymphoid cells, and natural killer cells. These differences in histone PTM patterns reflect the underlying variation in regulatory activity in chromatin states. And now that we've seen how CUT&RUN performs across different cell types and targets, let's take a look and see how CUT&RUN can also be used to investigate the inhibitor mechanisms. So we took H1299 cells and treated with 3 inhibitors for SMARCA2, a remodeler. The first compound, BRM014, is an allosteric inhibitor, and the remaining 2 compounds are PROTACs. And PROTACs are compounds that bind their target and mark it for degradation with ubiquitin using a fused E3 ligase. And once these cells were prepped, CUT&RUN was completed. So here we are looking at genome browser tracks and comparing CUT&RUN signal for histone modifications, as well as a non-remodeler target, INO80, and the remodeler target, SMARCA2. In the control condition and in the allosteric inhibitor, we can see strong signal for SMARCA2, whereas in the PROTAC-treated condition, we're seeing a peak loss and decreased peak enrichment, indicating the protein loss. I also want to highlight the signal for INO80, and that is the signal is not lost in the presence of any of the compounds, and this shows the specificity of the compounds to their target. So together, these data demonstrate how CUT&RUN can sensitively detect protein loss and distinct mechanisms of inhibitors. Now that we've seen examples of high-quality data sets generated using CUT&RUN, it's also important to acknowledge that there's challenges that can impact data quality. Like any technique, CUT&RUN has a few common pitfalls, and recognizing these issues early can help avoid failed experiments and improve reproducibility. For CUT&RUN, there are 2 areas which can jeopardize the experiment. First is the antibody selection. The common issue seen is that antibodies are unsuitable for the application, and there's a lot variability. A few ways to be aware and prevent issues is selecting distinct antibodies, examining positive and negative controls, and looking at the genomic distribution. The second pitfall is in the sample prep. And the source of the pitfall can be attributed to poor cell quality, too low cell numbers, and cell fixation. And the major way to overcome this challenge is the 3 major QC checks. And I will go further into detail in the next coming slides. With the right tools and guidance, generating your own high-quality data set is entirely achievable. And the protocol can be segmented into 5 sections. And for the remainder of our time together, I will navigate the workflow, sharing practical tips to help streamline and enhance the user experience. First, starting with reagent selection, antibodies, and controls. When we look at PTM antibodies versus CAPs, and I'll also go through the benefit of using a nucleosome spike-in and how to assess a successful target for novel targets. Next, we'll look into sample prep and look at the types of materials that can be used and those quality control checks. Then we'll move into day one, sample binding and chromatin targeting, looking at Con-A bead activation, target assignment, and sample binding to the solid support. We'll move into day two, which is categorized as chromatin release and purification. And we'll look at the PAG MNase activation and digestion, enriched DNA purification, and library prep. And then lastly, we'll go into the next generation sequencing and the data analysis. Since cut-and-run is an immune tethering approach, finding a suitable antibody is crucial for the experimental success and the biological interpretation during data analysis. So when you select your antibody, you want to be sure of its specificity. There are two major classes of targets, histone PTMs and CAPs. For histone PTMs, we recommend using our nucleosome spike-ins for determining specificity. And for CAPs or histone PTMs that have unavailable spike-in nucleosomes, we recommend using biological enrichment. So first, focusing on histone PTM antibodies. The most problematic concern is that the PTM antibody often recognizes off-target PTMs. And this is highlighted here, where over 300 antibodies were screened for their specificity using a nucleosome spike-in, and for over 70% of the antibodies failed. When the criteria for passing was only less than 20% cross-reactivity to a different PTM and capturing at least 5% of its target. However, good antibodies do exist, and in-application testing with spike-ins is capable of identifying highly specific PTM antibodies. The in-situ validation includes a panel of PTM-defined or epitope-tagged nucleosomes that are wrapped by a unique DNA barcode. These spike-ins are added to the cut-and-run workflow prior to the target antibody and is therefore processed alongside the sample chromatin. After sequencing, the read count of each spike-in nucleosome barcode provides a quantitative metric of on versus off-target recovery, therefore gauging experimental success, as well as a sample normalization tool for cross-sample comparisons. Here we are visualizing spike-in specificity for 7 different antibodies. Antibodies which only recover a spike-in nucleosome carrying the target PTM with less than 20% cross-reactivity to off-targets are considered a successful antibody. And this K-MET-STAT panel serves for a dual purpose. One, confirmation that the experiment that was run successfully, which is evident by the 3 controls, IgG, HCK4-trimethyl, and HCK27-trimethyl. And secondly, it can also be used to validate new reagents. And in this example, it's HCK36-dimethyl. In this data set, the x-axis displays all the PTM-defined nucleosomes within the panel that are available for capture, and the y-axis shows the antibodies tested. The coloring shows the on-target recovery for each PTM, where red is set to 100% on target, and the 3 controls, IgG, HCK4-trimethyl, and HCK27-trimethyl, and 1 HCK36-dimethyl antibodies are considered successful, whereas 3 HCK36-dimethyl antibodies from different vendors are considered failed due to their greater than 20% cross-reactivity to other histone PTMs. Selecting a target antibody for CAPs relies on biological enrichment. And for every new target, PTM or CAP, we recommend selecting 3 to 5 antibodies that are distinct, meaning they are created using different immunogens or from different host species. When to screen novel antibodies, starting with 500,000 cells or nuclei, since antibody efficiency can drop with lower cell numbers. Highly efficient antibodies maintain peak number and peak enrichment and has a high signal to noise even with fewer cells. The genomic distribution of peaks is consistent with the reported target function or is correlated with epigenetic marks, and it can overlap with other targets within the same complex. Prior to selecting a target, you want to ensure that the target is localized to chromatin and expressed in your cell of interest. And lastly, you want to select a reagent that has the best balance of the cut-and-run yield and high signal-to-noise ratio. One example of how these 5 recommendations are helpful when spike-in nucleosomes are unavailable is shown here. Multiple p53 antibodies were screened in 500,000 K562 cells. And of the 5 antibodies, only antibody number 1 generated the best signal with a strong peak structure associated with active transcription with a high correlation with HTK4 trimethyl and a low correlation with HTK27 trimethyl. Now, whether the target is a histone PTM or a CAP, some key metrics to keep in mind when selecting a reagent and your choice should be: the genomic distribution of the peaks is consistent with the reported function, 2 or more independent reagents yield highly similar peak locations, and then your positive and negative controls perform as expected. And your negative control is IgG, which should have a low genomic enrichment, your HTK4 trimethyl, which should have punctate narrow enrichment associated with promoters, and HTK27 trimethyl, which should have broad genomic enrichment associated with repressed regions. When your control antibodies generate the expected spike in recoveries and the genomic features, the cut-and-run data is reliable and the workflow is validated. The controls inform the user of failed reactions, samples, or the entire workflow. Now, the second major variable within the cut-and-run workflow is sample prep. Cut-and-run is compatible with a range of cellular material, including suspension or adherent cells, cryopreserved cells, tissues, or primary cells. Some considerations to keep in mind is cut-and-run is not suitable for heavy cross-linking, but is suitable for native or lightly cross-linked cells or nuclei. Always start with 500,000 cells per reaction. However, high-quality antibodies can be successful with fewer cells. Optimal samples are monodispersed with minimal cell lysis and normal morphology. And nuclei extraction is recommended when working with immune cells, since Con-A is a lectin and can be immunostimulatory. And nuclei extraction is often required for tissues and neurons. Most users begin with a cell line model, since these are easy to grow and handle, but there is a growing interest in adapting cut-and-run for more complex samples like tissues. But we're going to start with the recommended workflows for cells to generate cut-and-run-ready samples. Adherent cells can use a light trypsin digestion to help generate a suspension cells. Then all suspension cells can be collected and centrifuged and washed in the cut-and-run wash buffer. If light fixation is required, the harvested cells should be resuspended in a 0.1% formaldehyde, and after a brief 1-minute incubation, glycine is added to quench the fixation. Cells are then washed one time in a complete growth media and centrifuged and resuspended in the cut-and-run wash buffer. Cryopreserved cells are thawed at 37 degrees C, resuspended and centrifuged to remove the cryopreservation media, and then resuspended in the cut-and-run wash buffer. Once the cells are resuspended in the cut-and-run wash buffer, they can be counted and assessed for quality under the microscope. However, all of these cellular materials are also applicable for nuclei extraction workflow, where the harvested material is then resuspended in nuclei extraction buffer instead of cut-and-run wash buffer. And once the cells are resuspended in nuclei extraction buffer, they go through a short incubation, 10 minutes on ice, and afterwards the cells are then centrifuged and resuspended in fresh nuclei extraction buffer. These sample processing workflows give you the flexibility to process a variety of sample types. So whichever processing methodology will fit your experiment needs, we recommend processing an additional 25 to 50% excess. This is to account for loss during washes, centrifugation, and nuclei extraction. Preparing tissues for cut-and-run is not trivial, and we have the following recommendations for fresh versus frozen tissues. So first, processing fresh tissues, the samples will begin with the tissue mincing and enzymatic digestion to generate a single cell suspension. And once the cells are collected and centrifuged, the single cell suspension can be washed, resuspended, and cut-and-run wash buffer. And once the fresh tissues are resuspended in that cut-and-run wash buffer, they can be counted and assessed for quality under the microscope. Whereas frozen tissues are processed through a tissue dissociation, followed by centrifugation to remove debris, and the cells are then resuspended and prepared for nuclei extraction, which will follow the same nuclei extraction workflow to cell lines. Fresh tissues can also be processed by nuclei extraction. If desired, instead of resuspending in the wash buffer, you would then just resuspend in the nuclei extraction buffer. Additional cleanup may be necessary, and sample prep should be optimized for each tissue type. But whether you're starting from native, cryopreserved, or tissues, each type can be processed in a way that maintains data quality and reproducibility. And now, once all the sample material and the workflow is selected, we can discuss assessing their quality. Counting, examining sample integrity, and permeabilization is checked using trypan blue exclusion. Using a bright field microscope or a cell counter, dead cells will stain blue, and cells with an intact membrane will exclude the dye. When using fresh material, aim for greater than 90% viability. However, cryopreserved and compound treated material may have increased permeability, so therefore you want to focus on total counts, cellular morphology, and the amount of debris present. Move forward with cut and run if the debris is minimal and the majority of the cells appear normal. Now, nuclei will stay positive for trypan blue and will have intact nuclear membranes. Disrupted nuclei will have disrupted membranes, which indicates a poor quality material and will yield poor quality cut-and-run data if this is the predominant species. Now, during sample prep, in the early steps of cut and run, we recommend checking samples at 3 time points: initial cell harvest, post-washes, and post-binding to CON-A. The first is during the initial cell harvest, where you want the cells to have little cellular debris and a normal morphology. The second is after the wash in the cut and run wash buffer. The cells should maintain their viability and the quality they had during the initial harvest. Or if you're working with nuclei, the second check is after nuclei extraction. This check visualizes the nuclei, where the nuclei should be trypan blue positive and have their intact nuclear membranes. The third QC check is during the CON-A bead binding, where images are taken at 2 points. The first is after binding by checking the supernatant of the unbound fraction, which should be clear of cells or nuclei and the beads. The second checkpoint occurs when the cells are resuspended in the antibody buffer. At this stage, we recommend taking a small aliquot of the sample, ideally using a high abundance target such as H2K37 trimethyl, or running an additional QC reaction. And in this check, the cells and nuclei should be surrounded by the CON-A beads. CON-A beads are in excess, so the floating beads will be present and is not indicative of a failed binding. So once the target reagents have been selected and the samples processed, cut and run day 1 can be established. And during day 1 of the workflow, CON-A beads are activated through a series of washes to remove the CON-A from their storage buffer. If CON-A beads are damaged or dried out, the cut and run will fail. Signs of CON-A bead damage includes clumps after mixing, granular or grainy in texture, and I have this displayed here, or they appear black in color, and this can happen if the beads are frozen. Spiking nucleosomes are added prior to the target antibody to each reaction receiving a histone PTM or an epitope tagged antibody, and the next is adding the primary antibody. We recommend adding 0.5 micrograms or the manufacturer's recommendation of the target-specific antibody to each reaction. And at 500,000 cells, 0.5 microgram is in excess, and adding more antibody can increase the background. Each reaction only receives 1 target-specific antibody. The next, you place the tubes for an overnight 4 degrees C incubation with elevated caps at a 45-degree angle on a nutator. If placed on an end-over-end rotator, the CON-A beads will stick to the lids and dry out, resulting in sample loss. And this will wrap up day 1 of the cut and run protocol, where we have completed sample prep, CON-A, and antibody binding. On day 2 of the cut and run, it can be broken into 3 sections: chromatin digestion, DNA purification, and library preparation. During chromatin digestions, reactions are washed to remove the excess primary antibody. CON-A beads can often become clumpy after the overnight incubation, and some clumping is normal and should not impact the data. However, excessive clumping can result from poor sample quality and can lead to sample loss and reduced DNA yields, which will overall negatively impact the data quality. Gentle pipetting or vortexing can disperse the beads. Next, PAG emanase is introduced into the workflow and is used to cleave and release the antibody-bound chromatin fragments. Through our team's research and development efforts, we have identified the optimal temperature and incubation time for PAG emanase binding and activation, just at 2 hours at 4 degrees C. Extended incubations or elevated temperatures can lead to overdigestion, while insufficient time may result in underdigestion. Quenching emanase is crucial to obtaining the mononucleosomal fragments. The stop buffer, which chelates the free calcium ions to quench the PAG emanase activity. And during the stop buffer addition, you can spike in E. coli DNA, which can be used to monitor library prep and be used as a normalization tool for analysis. You want to aim for 1% E. coli in the total sequencing reads. Low abundance targets can have upwards to 10 to 20% E. coli reads, whereas high abundance targets can have around 0.1 to 1% E. coli reads. E. coli percentages outside these usual ranges can often indicate a poor cut and run reaction. All the cellular material remains on the beads, so it is crucial to transfer the supernatant to new clean tubes for the next step. DNA purification. Many DNA purification approaches are compatible with cut and run, including spin columns or magnetic beads. We recommend the CUTANA DNA purification beads to streamline purifications in the H-strip tubes and for efficiently capturing your enriched cut and run DNA. Bead ratios can be adjusted to influence the fragment size selection. For example, increasing the ratio will favor the capture of smaller DNA fragments. And then during elution, you want to ensure that the beads appear a matte and dark brown. If the beads are wet and appearing glossy, or dried out appearing cracked, it will negatively impact your DNA yield. DNA yields can be quantified by colorimetric assays. Low DNA yields may not reflect cut and run failure, since it is influenced by the target abundance, cell numbers, and antibody efficiency. Following DNA purification, the cut and run enriched fragments need to be converted to sequencing-ready libraries through adapter ligation and amplification. The library prep stage is important for preserving DNA integrity and the complexity for downstream sequencing. And during library prep, the cut and run DNA is barcoded and prepared for next generation sequencing. If DNA yields fall below 5 nanograms, we recommend using the sample in its entirety. Now, various commercial kits are available and will include all the necessary reagents to create distinguishable libraries. Regardless of which kit you use, we recommend following EpiCypher's cut and run specific PCR parameters, which have been optimized for low inputs in the small DNA fragments, specifically the 200 to 700 base pairs. If yields are low due to, for example, low cell inputs, PCR cycles can be increased to 16 to 18 cycles. However, if you're starting from less complexity, the reduplication rate in your sequencing data will likely increase. And within library prep, it is completely unnecessary to shear or fragment the DNA. Library yields are not the only success metric, since yields are influenced by target abundance, antibody efficiency, cell type, and number. If your yields are less than 1 nanomolar, consider repeating library prep with more starting material. Alternatively, you can add as much as the lower concentration libraries to the sequencing pool, but deeper sequencing, meaning over greater than 10 million reads, may be necessary to capture the read diversity. One way to predict cut and run success before sequencing is assessing the fragment distribution of the purified libraries. When analyzed on a bioanalyzer or a tape station, shown here, a successful library will typically show a primary peak around 300 base pairs. However, one thing to watch before loading cut and run libraries onto the sequencer is the adapter dimers, and they show up as small fragments around a peak between 100 and 175 base pairs, usually around 120 base pairs, and this will eat up all your sequencing reads. Adapter dimers will need to be removed prior to sequencing if they comprise greater than 5% of the total DNA. But a quick size selection, bead cleanup, or gel excision can be performed once all the sequencing pools are pulled together. This will help exclude adapter dimers to ensure a high quality sequencing data. Cut and Run is compatible for multiple sequencing platforms, and we recommend choosing a platform that utilizes paired end sequencing, since this improves genomic mapping accuracy and distinguishes the nucleosome spike-ins. Cut and Run also only requires the 5 to 10 million total reads per reaction to create the appropriate peaks and cover the genome. And then during Cut and Run, the same scaled amount of the nucleosome spike-ins and E. coli DNA is added to the related reactions, and therefore can be used as a normalization tool to scale the signals across the reactions. And once the libraries are loaded onto the sequencer of choice, the run is complete. And then the next step is moving into the computational phase of the workflow. This is where the bioinformatics pipeline takes raw sequencing reads and transforms them into accessible data for visualization and analysis. And this pipeline is segmented into 4 steps. Step 1 is demultiplexing reactions, where each reaction is pre-processed, adapters are trimmed, and the FASTQs are generated. Step 2 is the genome alignment. We recommend using Bowtie 2, and here are a few examples of available genome builds: HD38, MM10, and T to T. Step 3 is the primary analysis, where each reaction gets multi-aligned reads, duplicates, and the exclusion list removed. Also in this step, the nucleosome spike and reads are recovered. And step 4 is the secondary analysis, and this is where the reactions are filtered to create files for visualization, such as the genome browser tracks I shared earlier, as well as creating files for genome annotations in the motif analysis. To illustrate the secondary analysis possible, here are a few possibilities that can be used to deepen your insights from your cut-and-run data. And this data was generated using a target-specific antibody to the pioneer transcription factor, FOXA1. So starting on the left, we can see heat maps showing signal of enrichment and centered around peaks called for FOXA1. And there's strong enrichment for FOXA1 and a low background signal at these peak locations, for where over 6,000 peaks were called. Next is the bar plot that shows the genomic distribution of the FOXA1 binding sites. Most of these peaks were called in the intron and intergenic regions. Next, we can see the overlap between FOXA1 called peaks and the canonical FOXA1 motif. And this shows that the cut-and-run is capturing biologically relevant sites. And lastly, we see the genome browser tracks of FOXA1 called peaks, where FOXA1 displays sharp, distinct peaks, which are absent in the IgG control. And then within the FOXA1 peaks, we can find the canonical motif. And this FOXA1 example showcases some of the secondary analysis possible that extends beyond just peak calling. So bringing it all together, and to ensure you leave with a clear understanding, here's a recap of what we covered. Cut&RUN is a powerful tool for high-resolution and chromatin profiling, which needs lower cell inputs and lower sequencing needs compared to ChIP. It's compatible with more diverse targets. The cut&run reagent selection requires evaluation. Nucleosome spike-ins are instrumental, especially for histone PTMs. Cap targets should have a distinct genomic enrichment, and you always want to include your negative IgG and your positive controls, H3K4 trimethyl and H3K27 trimethyl. Cut&run is compatible with diverse sample formats. Cells or nuclei are compatible with cut&run, and nuclei extraction is required for tissues and is recommended with immune cells. And heavy cross-linking is not suitable for cut&run. Lastly, the cut&run bioinformatics deciphers the epigenetic landscape, where individual reactions are identified by their unique barcodes, reactions are aligned to the genome of choice, and reactions are processed for secondary analysis, such as peak calling and motif enrichments. Now, I just want to take some time to quickly highlight a couple of opportunities that might be of interest. If you're planning to attend EpiCypher 2025, registration is still open, and this is a great way to connect with the community and learn about the latest developments in chromatin in the epigenetic fields. Also, keep an eye out on your inbox. We'll be sending out exclusive offers by email following today's session. And then if you're looking for additional guidance or troubleshooting help, we encourage you to check out the EpiCypher Tech Support Center. It's a searchable hub that's filled with articles, tips, and protocol recommendations for cut-and-run, cut-and-tag, and more. And then lastly, I just wanted to take a moment to share some exciting news that we will be launching a new service designed to support your data analysis for cut-and-run and cut-and-tag. We know that interpreting the data can be one of the most challenging parts of the workflow. So our upcoming service is built to streamline that process. This offer will help you move from raw data to interpretable results, such as analyzing the cut-and-run and cut-and-tag data, the FastQ alignment, generating QC report, and have the on-platform IGV browser to create those genome browser tracks. More details, including how to get started and when this will happen, will be shared closer to the launch. But we are really excited about this and how it can support your research. And at this time, I'd like to thank all the team members of EpiCypher for their contributions, as well as our collaborators. This project and more was supported by the NIH, and we are grateful for their continued investment. Thank you all for your attention, and I'd be happy to answer any questions.
All right, Tessa, thanks so much for that. It looks like we have a ton of questions that were added in the chat. I think our colleagues got to many of them, but I'll go ahead and read some aloud, if you don't mind. And any questions we don't answer, we'll definitely reach out via email. So the first question, 3 to 5 distinct antibodies to test for cut&run. Could you reiterate our antibody testing regime that we usually employ?
Yeah, we usually recommend 3 to 5 different, so usually that are antibodies that are picked from like different immunogen regions, like different like amino acid, like 1 through 30 versus like 103 to like 400, depending how big your protein is. Or if they're from the same immunogen region, try looking at different species, so like a rabbit versus a mouse antibody, or like a squeeze.
Okay.
It is.
All right, so we get another question here. Can I isolate DNA structures if an antibody recognizes them, or do I need to cross-link the protein?
DNA structures, like a whole, like a complex, or like what type of DNA structure?
Yeah, I would assume, I guess just kind of maybe reiterate our kind of testing regime when it comes to native versus fixed for a lot of like TFs and chromatin-associated proteins.
Sure. So yeah, if you're thinking cross-linking might be something you would want to do, we always recommend running in parallel your cell of interest with your targets first with native and cross-linking conditions at the same time. That way you can actually, when you look at the data, you can see if the cross-linking condition actually preserved your mark or your complex structure before doing any future studies. So running them in parallel at first.
Okay, another question. How do I minimize cell loss? I'm sorting LSK cells from bone marrow, which are limited to 50K to 100K cells.
How to minimize cell loss?
Yeah, I guess like, would you focus on selecting those cells that are live versus dead? Or
Yeah, well, your permeability can change, so you would really just want to make sure that your morphology is intact and like you have very little like cellular debris present, because once cells are sorted or if they're frozen or treated, their permeability changes, but they're still viable and can be used for cutting.
Okay, I've got another one. I think I could probably answer this. So how important is the 45-degree angle for overnight shaking? We're unable to do this on our Hamilton. So I assume they're using a liquid handler Hamilton instrument. So we've actually done a lot of auto cut and run projects, including Hamiltons and TCAN as well. So we'd be happy to have a further conversation about, you know, what settings we would recommend for robotic handling of the cut and run assay. All right, so another question here. Adding more cells than 500K can result in higher background. What do you think about that one?
It can, I would say maybe for like your IgG targets specifically, because your beads are in excess. So the more cells you add, the more like you're going to have. 500,000 cells for most cells is plenty. Necessarily adding more isn't going to help your cut and run data.
Yeah, I think the maximum that people have had success with is about 1 million. But 500K is usually the starting point that we recommend. So let's see, another question here. What computational package do you use to generate heat maps?
We often use deep tools.
Deep tools, yeah. Yeah, so we can definitely follow up with that particular individual about the tools we recommend for doing computational analysis of cut and run. Let's see. There's a ton of questions. So what about how common is it to under or over digest? Is that a problem that you've, I guess, encountered before?
I don't. If you do the 2 hours like 4C during the actual activation, I don't think you will encounter that over digestion or under digestion. But if you were to like leave your samples at room temperature, then you can definitely probably start to see that over digestion occur.
Okay, another one here. So this question's about qPCR and cut and run. What are your feelings regarding qPCR and cut and run?
You do not need to do qPCR. qPCR was used to confirm enrichment in ChIP, which uses like the bulk chromatin as an input, but cut and run uses the cells, so it's unnecessary to use qPCR. And usually your yields are going to be too low for the input anyways.
Okay. So do you recommend doing a tape station following the cut and run assay?
No, you would only want to do tape station after library prep, because again, your yields right after cut and run, when you just enrich them, is going to be too low for like the tape station. But you can use something like a qubit fluorometric assay or even like a pico green.
Okay. So someone else asks, did you say that you could add 10 to 50% more cells to account for cell loss?
That is, so when you are like harvesting your cells, you would want to harvest like that 25 to 50% excess. Like say if you're going to nuclei extraction and you need like 5 million, I would maybe harvest like 7 million, do the nuclei extraction, and then make sure each reaction is still only getting about 500,000.
Okay. So what QC experiments should we do before sequencing? It's a pretty common question.
Definitely look at your samples, like the initial cell harvest. If you're doing nuclei extraction, post-nuclei extraction, the post-washes. Definitely look at your unbound and your sample slurry from the Con 8 bead binding. I would also check your DNA yields with like the fluorometric assay, and then before sequencing, looking at the tape station traces is really important. Especially right before you load it onto the sequencer, I would look at the individual concentrations, then looking at your pooled library. When you dilute it down for the sequencing platform you're using, I would check it then as well to make sure what you're loading is what you think you're loading, and using the tape station for all those steps before you actually load it onto the sequencer.
Okay, so do you have a protocol to isolate tissue? I guess cells from tissue or nuclei?
Cells from tissue. So we don't have like a published protocol exactly, but on the tech support page that I showed briefly, you can find some links to various papers that have methods, or if your lab has a method, go ahead and give that a try. But the overall kind of basic steps that would include is the tissue collection, the disruption, the enzymatic, the digestion, or the grinding of them, nuclei extraction, using our nuclei extraction buffer, and then once you have those isolated nuclei, going into cut-and-run.
Okay. How do I know if I should cross-link cells?
So cross-linking can be helpful for labile marks, for like acetylation or ubiquitination, but for any time I think cross-linking is in consideration, I would run native samples alongside with a cross-linked set to see which one is actually going to be suitable for you.
Okay, so here's a good one. So what is the average DNA size post-cut-and-run for histone marks in TFs?
So TFs will definitely can differ a little bit because some transcription factors have like a smaller DNA motif, or some can be part of a larger complex. So that's a little bit shifted. But for histone PTMs, for the tape station, you're really going to see that 300 base pairs, which is the 147 mononucleosome fragments with the adapters added onto it. Okay. So I would still say around the 300 base pair peak is what you're still looking for, no matter the target.
It's a pretty common one. So what does a bad Con A binding look like?
I would say bad Con A binding is that your unbound fraction, you're going to see a lot of cells floating. And if you have maybe over 50% of your cells in your unbound fraction, that's going to be a sign of a bad binding. And then in your sample slurry, you're not going to see them like being like attached or having contact of the membranes of your cells or nuclei.
Okay. So here's kind of an interesting one. So for transcription factors with low cell number to start, somewhere between 50 and 100K, what do you recommend to do? Cut and run or cut and tag?
For transcription factors?
Yeah. So why do you think?
Well, so the differences between like the cut and run protocol and the cut and tag protocol is that cut and tag during the TN5 is going to use a higher concentration of salt, and a lot of times that salt is high enough to disrupt the contact of your transcription factor of interest with the chromatin. So you actually end up losing some of that. Whereas cut and run is at a lower salt, and it's maintained throughout the course. So you actually will be able to get more of that DNA out. So we always recommend cut and run.
Yeah, cut and runs are very do-it-all kind of assay, especially with transcription factors.
Yeah, and if you have highly efficient antibodies, you can titrate down. You don't need to do it 500,000 cells.
That's right. So what are normal yields of cut and run DNA that you get from the positive control antibodies? For example, the K4 tri that we include in our kits, using 500,000 cells as input. I've gotten 3 to 5 nanograms. Is this normal?
It's going to depend, I feel like, on your cell type, but I think we like over 2 nanograms for me is what I've gotten commonly over K4. So 3 nanograms would be normal because it's going to depend on your cell type and your cell number.
So I guess, is it common to have like too low to identify from qubit, and what do you do if you have that result?
Yeah, if you have too low of yields from like your qubit, I would just use your entire sample for the cut and run library prep. Just use it all. Most times you still have sufficient library. You can make a sufficient library even when it says it's too low for the fluorometric assay.
Okay.
And then really look at the tape station trace. If it has that normal peak, continue to sequencing.
Yeah, so just if you have too low to quantify yields, like not all hope is lost. So how much clumping is normal in cut and run?
Some clumping is normal. You'll definitely see it, especially overnight. You can kind of see that settling of the beads when you come in the next day. Usually just gentle pipette mixing or vortexing will resolve it. If gentle mixing doesn't, then you might experience some concern, but it is normal part of the assay.
Right. So do you add the same amount of DNA for all samples when preparing libraries? That's a good question.
Yeah, if you can, I would do like 5 nanograms is what we recommend. But if it's below the 5 nanograms, then use it in its entirety. I wouldn't add more than 5.
Okay. So I have a low DNA yield after cut and run. Should I increase my PCR cycle number?
I would recommend no. Start first with like 14 cycles, because usually most times the 14 cycles will be enough to amplify your library. Just because like, increasing the libraries will also increase your duplication rate. So I would say try first at 14 cycles before just upping to 16 or 18.
Okay. Yeah, so I know our early kits had column purification, but now we've kind of moved to a spry method. So what ratio, I guess, do you typically use? And I know we just came out with our own version of spry beads as well.
I think we currently would recommend like 1.4 to 1.8x, depending if you're 1.4 for like most histone PTMs, and then if you are looking for a smaller fragment, increase up to 1.8x to capture it.
Okay. How do I freeze my cells? Another question.
So cells can be frozen in a freezing media, which is like 90% full like growth media and like a 10% DMSO. And then they are slow froze in like an isopropanol chiller at -80, or you can keep them stored in like liquid nitrogen and then just thaw when you're ready to use them.
So do you recommend snap freezing or just like a slow freezing with the method you described here?
The slow freezing will work just fine.
Okay. Let's see. Can I do single cell? What about FFPE tissue?
So first, note for the FFPE, because FFPE tissues are heavily cross-linked, and heavily cross-linking is not suitable for cut and run. You can only go up to like light to moderate cross-linking. Single cell, I don't think it's been done for any cut and run. The lowest input we have seen is down to like 5,000 cells per target. And since it's really dependent on like, the antibody efficiency and how to create that single cell like library for complexity, it's just not really in the cut and run technology. So not applicable for either.
Here's a common question we get in tech support. So what do Con-A beads look like if they're bad?
So they will appear like black in color if they're frozen, or they can be really like gritty, almost like coffee grounds, and they will clump together if you try to mix them, and they'll like clump together and settle out of solution if they're damaged.
So it's pretty readily identifiable, right?
Yeah.
All right, maybe a couple more questions here. So what steps do you recommend for making sure that I load enough pooled libraries onto the flow cell or sequencing cartridge?
So if you want to make sure you load enough, one technique you can do is to first reverse pipetting. This will also ensure that you don't add any like bubbles into the cartridge, and then to make sure you're loading enough of your libraries is just really checking with the tape station or bio analyzer, looking at those fragments and concentrations, and looking at your sequencing pool, and then looking at the pooled libraries all together post and after you need adapter dimer removal. Quantifying on the tape station.
Okay. All right, so last question here. Again, we're going to answer any question we didn't get to via email. But would repressive histone marks be harder to target than active ones due to accessibility?
We have been able to map a wide range of targets from K9 trimethyl, which is really heterochromatin, and like K27 trimethyl. We've all been able to map those in cut and run.
Yeah, so there's really no limit between facultative heterochromatin and constitutive heterochromatin in terms of cut and run. All right, well, it looks like we've answered a ton of questions. Thank you so much, Tessa, and the tech support team. To learn more about our assays, reagents, services, please check out our recently updated website. We've got a categorized product table on there and an improved search function. Of course, we'll be happy to see all of you at EpiCypher conference in Cancun in November. So the rooms are limited, so please register soon if you're interested in that. And thank you all. Have an amazing day.